Good afternoon, everyone. This is Amalia from GenScript. Welcome to our next presentation on this GC Summit. It is my great pleasure to introduce our speaker, Dr. Fred Parietti. Dr. Parietti is the co-founder and CEO of Multiply Labs, a robotics company developing industry-leading automated manufacturing systems to produce individualized drugs. Founded in 2016, the company's mission is to scale patient access to cell therapies and other individualized medicines. Under Dr. Parietti's leadership, the company created the first cell therapy robotic system to be compatible with existing market-leading GMP equipment. This means cell therapy manufacturers can scale without significantly changing their existing processes, minimizing process and regulatory risk. Additionally, Multiply Labs brought to market the first technology to manufacture individualized pharmaceutical capsules at industrial scale. Dr. Parietti holds a Ph.D. in mechanical engineering from MIT. He has co-authored over 30 peer-reviewed robotics publications and has over 12 patents and patent applications, which have been cited over 2,000 times. Prior to his research at MIT, Dr. Parietti worked on robotics at Carnegie Mellon University, ETH Zurich, and Polytechnic University of Milan. For today's presentation entitled The Power of Automation, How Robotics Can Dramatically Reduce Cell Therapy Manufacturing Costs and Scale Patient Access, Dr. Parietti will share with us how harnessing robotics and automation within the cell therapy space can impact the economics and throughput of the manufacturing process, as well as how it can significantly accelerate timelines and reduce regulatory barriers and human errors, resulting in expanded patient access to life-saving therapies. As a reminder, if you have any questions, please input them in the Q&A box, and we will address the questions after his presentation. Without further delay, let's get started. Dr. Parietti, go ahead.
Thank you very much. Thank you very much. It's great to be here. I'm very grateful to the GenScript team for inviting me to this talk, and I'm looking forward to the questions at the end of the talk. So let's go. So in this presentation, I will discuss the technology that Multiply Labs and a set of partners, including GenScript, are developing together in order to automate the manufacturing of cell therapies. And so let's start from the goal. Why are we doing this? We're doing this because we believe that essentially manufacturing gene-modified cell therapies at scale, it's possible substantially only by adopting advanced automation. We believe it's not going to be possible to manufacture enough cell therapies for all the patients that need them by using essentially legacy manual processes. It's also very important that we are focusing on the broad space of gene-modified cell therapies. So we did not develop this technology as just, a CAR-T technology or just an autologous technology. What we see as Multiply Labs is that in the broad space of cell therapies, including, a wide range, basically of cells, edited, with viral methods, non-viral, immune cells, other cells, iPSCs, all of these processes tend to have similar requirements and to use similar equipment. And so we really wanted to develop a technology that would be broadly applicable to basically the whole space. And so how do we do this? Well, this is going to be discussed in the rest of the presentation. So first, a kind of like an overview of the technology. I'm going to go much more in detail about this in the rest of the presentation, but there are 4 areas that we really focused on. One is the compatibility. Compatibility means that we do not design new instruments or new consumables or new reagents. Instead, we partner with the leaders in the space, including GenScript, right? And we teach our robots to use their instruments and their reagents and their consumables, right? So this is absolutely important. It's a core tenet of our approach. We don't force cell therapy developers to change their process. We adapt to it. We adapt the robots to it. This makes us very flexible. You're going to see it's a very modular technology. You're not, committing to a gigantic robotic system that can never, ever be changed. It's future-proof, meaning that we're always adding new instruments to the list of, the technologies that our robots are compatible with. And obviously cost-efficient. I would say the cost efficiency part, it's a common trait of all well-designed, I would say, robotic approaches. So the difference here is not the cost efficiency, it's how do you get there? What is the most seamless way to get to cost-efficient automation? Okay, how does it work, right? How can a robotic system operate an existing instrument? And this is kind of like the overview. We take the instrument, you can see here, example instrument, GMP ready with, extensive, GMP documentation and regular, and basically used in GMP. We take that instrument and we don't change it at all. The instrument is the same. We just put it inside what's called a robotic module, which is a cube. You can imagine a cube of roughly 3 feet in side length. And that cube contains the instrument itself and a set of auxiliary systems that enable our robots to operate that instrument. And then, of course, we have robotic arms that click in and out, consumables and reagents out of that instrument. And sometimes we need to repackage existing consumables within cartridges because robots really like rigid structured things. But in the end, the instrument and the consumable and the reagents are the same, right? Sometimes we add extra stuff around them, like to help the robots manipulate them. But from the sales point of view, nothing is changing. And this is really a powerful part of our approach. We have a similar approach on the software side, where we talk to the instruments, but we never override or modify their software. Obviously, this only works if we are covering or offering, let's say, integration, right, with enough instruments that most leading cell therapy developers would recognize the instruments that they're using, right? And so here you can see kind of an overview of what we have announced so far. There are more announcements coming up, but I can't talk about them yet. There is, in fact, a rolling list of new instruments that we are considering automating or actually working on automating already. And that's purely determined on the demand by the cell therapy developers, right? If cell therapy developers really want a particular instrument, then we're going to prioritize it and teach our robots to use it, right? So you can see this is already, an end-to-end system, right? And what we're going to do, is adding more and more, right, to make it even, more end-to-end to follow even more processes. But typically, if an instrument is not used in GMP and not widely adopted, we're actually not that interested in automating it because the whole point of the approach is to automate existing GMP processes, right? And so the demand from the point of view of the developer is very important. And you can see here our collaboration with GenScript is around enrichment and isolation, right? And in particular, the cytosync. This is something that we just announced, I think, about a month ago. It's a very exciting process. It's a very exciting module for which, we're seeing, a lot of interest, a lot of demand, because, of course, magnetic bead-based isolation is, I would say, kind of like the gold standard right now for most of the therapies we see. Okay, so every one of these instruments will go into its own module, right? And so you see the cube we were talking about before. Each of them contains a different instrument, and some of them we've announced, some of them are still confidential. When an instrument is big or when it requires a lot of supporting systems or materials, right, we also have the option to have a double height module. Like typically, imagine the incubator. We use Thermo Fisher incubators in this case, and essentially, you can imagine it. If you've seen some of them, they're pretty tall, right? And so they need more space, and that's not a problem. The robots can adapt to that. So every module is actually highly standardized. We don't really customize modules, right? Because every module is optimized for its own instrument, right? For example, the cytosync module is optimized for the cytosync. What is completely customizable is the combination of the modules. So you're going to see it here in this slide. So essentially, every collaborator, every partner can pick any combination of modules they want, right? So our hardware and software don't really care. They can adapt to any combination of modules. So if you're not using an instrument, you don't need to pick it, right? Or if you want to use a new instrument, you can add a new module. Most customers actually leave a couple of blank slots just for future expansion, right? And this really is powerful because it makes us very future-proof. So you have kind of like the confidence. You're not committing to a giant, unchangeable robotic system. As your process evolves, the technology can evolve with it, right? Provided that we have automated the new instrument you're looking for. And so that's why we work very, very closely with our partners to make sure that the new instruments that are available in a module form, right, are exactly the ones that the market is requesting. And typically, it's very obvious. Nobody wants to take risks, in a regulated space, for advanced biological therapies like cell therapies, right? So people do really tend to converge, in our experience, on using a few tried and true instruments. And so it's very easy for us, just by tallying those requests, to see where the market is going and to focus basically our engineers to provide those instruments that are in high demand. So like I was saying before, basically it's very easy to swap in and out the instruments. We don't show this in this drawing, but they're actually small rails, in fact, inside the structure of the robotic system. So it's actually very easy to, basically swap them in and out. It takes just a few hours, and I'm not saying that this should be done every day in a few hours, right? Definitely, it's designed to be a GMP manufacturing system, and every time you modify the system, then you're gonna need to run a validation protocol, right? So, but definitely there is that possibility that changing a single module does not require to change the whole technology, right? So it's a very powerful flexibility, and typically what it does is it puts our customer, who are cell therapy developers or large CDMOs, in control, right, of the process, and in control of which instruments they want in their platform, right? We don't tell that to them. They tell that to us, and the robots adapt to it. So how does the manufacturing process take place? It's actually carried out, of course, by each instrument, but the connection between one unit operation and the next one is done by robotic arm on a rail, one or more. Honestly, we've seen a lot of configurations according to the throughput and the demand, basically the type of process and the throughput required by the customer. We've seen configurations ranging from a single robotic arm all the way to 4 robotic arms in a single cluster, right? And the cluster is the set of modules. None of that is a problem. The robotic arm is essentially a dynamic way to moving a cartridge containing a product or reagents from any module to any other module. So we don't really have conveyor belts, because a conveyor belt process would be fixed. The robotic arm essentially has access to any module at any time. And so it's very easy to program any new process, right? Provided you have all of the instruments you need, it's actually quite straightforward to just send different instructions to the robot. And we do have a quite simple scientist interface. So you can just drag and drop the different tasks and essentially tell the robots what to do without needing to code, right? It's all a no-code platform. So it's really designed for ease of use. Of course, because there are many slots for different modules, and because we don't have requirements for the positioning or for the presence of any one really given module, if there is a bottleneck unit operation, what our partners typically do is they add more copies of the same module. So you just add multiple modules, multiple copies of this lowest unit operation. So if that's expansion, which typically is the case, you're going to have as many incubators as possible side by side. For example, we've shown we can fit up to 18 G-Rex, 1 liter G-Rex incubator, bioreactors inside a single Thermo Fisher incubator. So, if you had 1 incubator module, that would be 18 products at the same time. If it was 2, it would be 36. It was 3, it would be 54. It really depends on the throughput, on the length, of the expansion unit operation, right? In some cases, expansion is not the bottleneck. Maybe it's the robotic arm, so that's when we add more of them. And so all of these can be done. Basically, we can exploit parallelism by adding more copies of the same bottlenecked, essentially, piece of hardware. So that's exactly what I was mentioning before, which is a serial process. If it was a conveyor belt, right, or if it was a serial machine, or system, essentially only one part of the machine would be used, and so the slowest part of the system, right, automatically determines your throughput. Everything else is basically sitting idle. But because we can have multiple products in parallel, right? So for example, up to 18 G-Rexes in a single incubator, then you can really, leverage parallelism to increase throughput, right? And the results of these are very compelling. Keep in mind that, this slide, that I'm showing right now is, I would say, an average, right? Processes are very, very different from, one to another. We've seen anything from just a few days all the way to a few weeks, right? I mean, there are some iPSC processes that take a few months, per dose, right? So I would say the numbers you're seeing here are probably representative of a, typical CAR-T process, right? Where we see that, usually people need to use a suite with, 1,000 to 2,000 square feet, right, of space, multiple operators, maybe over more shifts. These really shrunk dramatically with the robotic system, where you only need one operator really to load the system and to oversee it. And the robotic system, according to the number of modules, but it only takes, a few hundred square feet of space. And by the way, inside the robots are basically a ISO 7 environment, so they can be placed in an ISO 8, or we have even data showing that they maintain sterility when placed in a non-controlled environment. And that data, by the way, has been published. We published it on Cytotherapy, the ISCT journal, last March. That was a collaboration with UCSF. Right now, we're in collaboration with the Stanford GMP Center for Cell Therapy Manufacturing, and we expect to publish data about that collaboration as well soon. And so yeah, stay tuned. But yeah, so we've always been very transparent on the performance of the robots, and what we see is a reduction in COGS of around 70%. When you basically automate an existing process. So the process is not changing, it's just moving from being manual to the same process being automated. And we really see a massive reduction in cost. We, in fact, put those cost estimates inside our published journal paper. So the reviewer had asked for it. And so we're actually very happy to share, those estimates. And so please check it out. It's if you're more interested, it's it's in the supplementary material available to freely download. And I think it's, I would say, very, very solid reason really to adopt automation in terms of efficiency and throughput. We see an order of magnitude reduction in cost and 1 or 2 order of magnitude increase in basically number of products per square foot of production floor. So here you see in a normal suite, there is a lot of limits on how many products you can make at the same time, right? And then you risk deviations, you risk cross-contamination. In a robotic system, you can, we've now based on what we developed so far, 18 products in a single incubator are possible. So if you have 3-4 incubators at the same time, right, it's 18 times 3 or 4, right? So that's a level of density that's just not possible, manually. And of course, the robots are very robust. And so I wanted to conclude the talk by kind of giving you a sneak peek of something that we're just starting to announce and reveal in public. There should actually be an announcement out today, I think, an interesting story on a specialized industry outlet where essentially like you saw before, we are teaching the robots to execute a process that right now is very manual. And so for most of the tasks, it's enough to just teach the robot to use an existing instrument. But there are some tasks where they're not really carried out in an instrument. They're carried out by hand by a extremely highly trained, highly skilled operator. And typical tasks are, I would say, what I would call shaking tasks, like resuspension, like mixing, right? Those tasks always have, in our experience, very complex trajectories that are very poorly defined. They are typically left to the individual operator. So even within the same organization, 2 different operators will resuspend the cells in basically 2 different ways, sometimes slightly different, sometimes very different. And so we want to make that more systematic. So we are actually using essentially trajectory data recorded from human demonstration to automatically train the robots to replicate that trajectory. So this is part of our collaboration with the Stanford Laboratory for Cell and Gene Medicine. And so we expect to publish some results about that as well. And the idea, is really like it should be easy for the robots to learn a new process by a combination of human demonstration and using instruments that are already being used, for that process, right? So on the robotic side, this is very exciting because I would say the holy grail of robotics is, learning from human demonstration without coding. And I think this is a very compelling application of that sort of new AI. It's called imitation learning, basically, set of technologies. Okay, so this is it. And I think we have, about 10 minutes for questions. So looking forward to hearing them.
Thank you so much, Dr. Parietti, for this really exciting presentation. It's incredible, the adaptability of multiply system and its potential to really, really adapt to any sort of system. So as you mentioned, we're on the Q&A session. So let's get started. So, could you comment on what does Multiply's partner ecosystem look like, aside from GenScript or other of the instruments that you also showed in your presentation?
Yes. Thank you very much. This is very important. This is really central to our technology. Because we do not design new instruments, right, or new consumables or new reagent, it's very important for us to essentially partner with all the leading instrument, consumable, and reagent providers. So not mentioned in this presentation where, for example, some of our reagent partners like Akron Biotech, and we're gonna, announce a few more. But the idea is, if you are a cell therapy developer, right, and maybe you are in the preclinical stage, but maybe you might be in the clinical stage, maybe you might be in the commercial stage, right? So as multiple labs, I can't, go in a call with you and tell you, you know what, you should change that reagent because it doesn't work on the robots, right? Like, that would be crazy, right? Nobody would do basically such a big change in order to adopt automation. We believe automation only works if it basically does not get in the way, right? If it's an extra layer on top of the process that was usually developed for years, sometimes almost like a decade, right? Before people even get in touch with multiple labs to scale, right? And so from that point of view, our partners ecosystem is already at this point unprecedented. There's no other organization in the industry that has so many partners. But the idea is, it really works that way, that we're not really competing with our partners. They, because we don't make reagents, consumables, and instruments. And at the same time, they're not really competing with us because they're not in the robotics business. So it's a very, very good, I think, ecosystem where we all understand how important it is to bring robotics to processes that are already very established with minimal changes. So I think our partner ecosystem right now is about, I think, 8 or even 10 organizations. I'm not sure how many. I think we announced 8. And so, yeah, I guess stay tuned for more. But the idea is, if you know their name and if you're using them as a cell therapy developer, we should work with them. And if we're not, let us know, right? Because it's very important that we do.
Thank you. Another question from the audience is related to the footprint. What's the footprint for this system?
Essentially, it depends on the exact number of modules, but I can give you a good, reference. Every module is about 3 to 4 feet inside length. So you can imagine like a 3-foot, cube, 3-foot side length, right? So, for example, what you see in this picture here, we have 1, 2, 3, 4, 5 modules on the side. So that would be, I would say, 15 to 20 feet, right? And then a 3, 3, 3, 9 feet, let's say 10 feet in the other, direction, right? So we're talking about, let's say, conservatively 420 by 10, 400 square feet, footprint. And let's say you have, 3 incubators inside here, right? Each of them can make 18, doses at the same time if you're using a G-Rex, right? Just making an example, right? So in that case, we'll be talking about 54 simultaneous doses in a 400 square foot, basically footprint. So, and again, like, it really depends on you and your process and which modules you want and how many do you need. But I think this level of density is already probably 100 times higher than the number of products per square foot that you can make in a manual process.
Got it. Another question is, what, in your opinion, is the biggest challenge currently facing cell therapy?
I mean, there are 2 big challenges, right? One we can address, another one we cannot. The one that we cannot address is definitely, I mean, we see a giant clinical pipeline, of cell therapies, right? At the moment, I think 7 to 10 are currently approved. So definitely there is a huge need, and these therapies are being, transformational. And so they will, I think the hope of the whole space is that more and more therapies in more and more indications, not just, oncology or, blood cancer, right? I mean, we're seeing already with Vertex new exciting indications. And so the first challenge is, I would say, establishing the space as, clearly dominant branch of medicine by approving more therapies. And that is the job of the cell therapy developers, right? We can definitely help on the manufacturing side. The second challenge is, after and even before these therapies are approved, well, you need to make them, and you need to make thousands of them. I would say you need to make, it's already very clear for the approved indication that the market is in the hundreds of thousands, I would say, right? Especially globally, but even in the US, we're talking about a demand that pretty much anybody would agree is already, around or even above 100,000 patients. And, maybe a tenth of that can be manufactured today, right? And so in this challenge, we definitely can help. That's why we designed these robotic systems. For the therapies that are advancing rapidly in clinical trials, that are in GMP, right, or that are already commercial, what is the fastest way to increase throughput in a way that's repeatable, that's scalable, that doesn't modify what was already approved? And so that's why we designed this technology.
Got it. I think we have a few minutes left for another question. So could you expand on how on the imitation learning process that you described? So it's kind of mind-boggling how a robot can replicate movements that are, very unique to a person, especially when they're so complicated and they're required to grow things as sensitive as cells and ensuring cell viability.
Yeah, no, it's very true. It's very true. But I would say, in fact, I think the only way is to teach a robot to do it. These motions are basically very poorly defined. Like in your typical batch record, you will see resuspend. That's it, like a single word. That could mean a potentially infinite range of trajectories. So the way these trajectories are defined today is actually not defined. It's really in the expertise, almost the skill, right, of the operator / artisan, performing the motion. So we see like yields changing when people change operator, right? Because the trajectory cannot be completely replicated. There is already established technology to, I would say, learn to measure a trajectory from a person demonstrating it, right? Technologies like motion capture, and so we're already using the technology. But the hard part here is we don't just want to record it once and then always copy paste it, because people adapt to different volumes or different cell types or concentrations, right? And so the idea is collecting enough examples and then having a robotic learning algorithm that is smart enough to generalize, or I would say, let's say this, interpolate within the range of possible parameters specified by the examples. And so this is really the cutting edge of robotic technology. And definitely sounds, complicated, and it is, very exciting for us as engineers, but it's definitely much simpler to do that than to tell an engineer, oh, look at that scientist, and now try to hard code the motion of the robots by specifying individual joint motion in the robotic arm. That would actually be impossible. So we think it's both needed and really the only way forward to use imitation learning type approaches.
Got it. Thank you so much, Dr. Parietti. This concludes our session. And thank you to all the participants and our audience. Please don't forget to visit our booth in the exhibit hall and stick around for the next session. Thank you so much.
Thank you very much.